Hello students, welcome to EPG Pathshala. I am Dr. Nirupama Trihinpati from Institute of Liver and Biliary Sciences. Today we are going to discuss the biotherapy of cancer from paper Molecular Therapeutics. Main objectives of biotherapy of cancer. First, we should know about what is the cancer and what are the microenvironment of the cancer and how the different biotherapies can be used to treat the cancer. Biotherapies are the living cells or living substances or the manipulation of the cells. There is a correction of the genes which can be used as the biotherapies to treat the cancers. It includes the adoptive cell transfers like even the stem cell transfer, T cell transfer, dendritic cell transfer. It also includes the gene correction therapies. It also includes the even newly discovered molecules like microRNAs or the RNase corrections. However, the recent discoveries in the signaling pathways corrections or the gene corrections are still on the animal models or the laboratory experiments, limited to the laboratory experiments. What are the biotherapies? As I explained you in my objectives, that biotherapy is the treatment designed to stimulate or store the ability of body's natural immune system to fight infection or the cancer. Biological therapies or called the biotherapies is to treat different kinds of cancers such like developed in the different condition or the metastatic cancers. Biotherapies use the drugs also. These drugs, they change the way the cell works and help the body control and growth the primary liver cancers. Some seek out and destroy the cancer cells also, and others help the body to attack the cancers. So these all are biotherapies. There are cancer cells which escape the mechanisms of immune response. Like all cancer cells, they show the defective antigen presentation. Tumor associated antigens are not presented well by the cancer cells. Tumor also induce inhibitory checkpoint pathways against the effector T cell activity. So there is no effector T cell activity to reduce the tumor. There are many suppressor or T cells which are the infiltrating regulatory T cells in the tumor. There is myeloid derived suppressor cells which are present more often in the cancer. All these suppressive T cells, including the myeloid derived suppressor cells, secrete the immunosuppressive cytokines like IL-10, TGF-beta. As we know that the tumor mass also produce the interleukin-6 and VEGF. Those all mechanisms, when the cancer cells have the more and they escape from the more checkpoint pathways, then it is developing the cancerous mass or the tumor is growing in large. There are strategies to develop therapies. Could be the strategies? Strategies could be that to boost effector cells of innate immunity like NK or macrophages. You have to boost the adoptive immunity cells like T and B cells. We have to boost the other escape pathways like including apoptosis and apoptotic signaling, mitogen activated protein kinase signaling, microphthalmia associated transcription factors, and the CMP signaling. We have to do the correction of genes. Latest discoveries are saying that microRNAs or the silencing of the RNAs also used as the biotherapy for the cancer. However, the popular therapies are including the cell infusions or adoptive cell therapies, gene therapies, microRNA silencing, New signaling molecules are still under discovery and standardized in the animal models only. Now in this slide, we will understand where and why biotherapy is being used for cancer. In the cancer, it is advisable to use biotherapy as the adoptive therapy, not the single. However, in the few cancers, clinicians or the Ayurvedic people have used only the biotherapy. But whenever the side effects of cancer treatment like chemo or the radiation therapies are more, then it is biotherapy is advisable. Biotherapy can also be used on its own alone or with traditional treatments of cancer such as chemotherapy or radiation therapy. What are the commonly listed biotherapies for the cytokines? Immunotherapy or the biotherapy use the body's own immune system to treat the disease, to restore or boost the immune system. There are many therapies like the cytokine therapies, include the interferons and interleukins. It also includes the colony stimulating factors. It can be used as a biotherapy. These kill cancer cells or to slow the spread of disease or activate the immune system to fight the disease. However, factors, colony stimulating growth factors, are being used to these cause the bone marrow to make the more immune system cells and blood cells. What type of biotherapies are being used commonly? Mainly immunotherapies or monoclonal antibody therapies, cytokine therapies and vaccine therapies are come under the cellular therapies and they are being used. Adoptive cell therapies like the T cell adoptive therapy, dendritic cell therapy is also being tried out in many clinical trials. Latest is the gene therapy, correction of genes. This is being used in the laboratories or in the animal models. Now I will explain you here what are the monoclonal antibody therapy. Monoclonal antibodies are kind of proteins on the surface of some cells onto it. This may then trigger the body's immune system to attack and destroy these cells. These monoclonal antibodies present on the surface of cells, however, locks into like a key and lock story. When biotherapy targets specific cells, these monoclonal antibodies are also called as targeted therapy. There are many vaccines which can be used for different diseases like human papilloma virus vaccine. There is the Simplex T vaccine, Bacillus Guerin BCG vaccine. So Bacillus calmette Guerin BCG and the Provenance T cell vaccine is used commonly in the different diseases and for the vaccinations. However, there are different types of cancer vaccines which are being used like antigen vaccines, whole cell vaccines, dendritic cells vaccines. I will explain you each one by one. Antigen vaccines, these vaccines are made up from special proteins in cancer cells. They aim to stimulate your immune system to attack the cancer. Many scientists have worked out the genetic codes of many cancer cell proteins so they can make them in the lab in large quantities and can be used in the animal models and standardized for the clinical trials. Whole cell vaccines uses the whole cancer cell, not just a specific cell protein like antigen. In fact, it is a whole cancer cell to make the vaccines. Researchers use these vaccines from their own cancer cells or another person cancer cells that were grown in the laboratory, like cancer cell lines. These cancer cell lines are being standardized in different experimental models. Third is dendritic cell vaccines. Dendritic cells help the immune system to recognize and attack abnormal cells such as cancer cells. To make the vaccines, researchers have grown dendritic cells alongside the cancer cells in the lab. So vaccine, when they make and they grow, that they have also grown together the dendritic cells and the whole cell lysate. These dendritic cells loaded with the whole cell lysate antigen or the act as vaccine, which stimulate the immune system to attack the cancers. Fourth is DNA vaccines. These vaccines are made with bits of DNA from cancer cell. They can be injected into the body to make the cells of the immune system better at responding to or destroy the cancer cell. Another type of vaccines are anti-idiotype vaccines. These vaccines stimulate the body to make antibodies against cancer cells. There is gene therapy or the gene correction kind of therapies. Target or the objective of these gene therapies to transfer the anti-cancer gene for cancer gene therapy. To treat malignant tumors, gene therapy vectors use a specific therapeutic transgene which cause the death of cancer cell. Therapeutic transgenes, generally it was earlier in the non-specific vectors. In the non-specific vector, it was killing the normal cells also and it was inducing the toxicity. However, now the gene therapy is being done with the specific viral vectors for the treatment of cancer cells, and it specifically destroys only the cancer cells, not the normal cell. This all therapy or the gene therapy including how it works. Gene therapy works, it induces the immune system, boosts immune system. Some types of gene therapy aim to boost the body's natural ability to attack cancer cells. These DNA or gene therapy specifically make neutralizing antibodies and increase CD8 T cell responses. Activation of myeloid or endothelial cell also does by the gene therapies. These gene therapies or DNA therapies supports T cell expansion, migration of these T cells to the tumor microenvironment. Gene therapies to make treatments work better. These are long-acting, slow effective release of antiretroviral therapies. It also mobilizes on their own myeloid-derived suppressor cell in the microenvironment of the tumors. These are tumor cell-based or the cell gene therapies. It, along with the vaccination, it induces the immunity. You need to know that why the biotherapy is being used for cancer. Biotherapies can treat the cancer itself, or it can treat the side effect of standard cancer treatment like chemotherapies or radiations. The treatment work in different ways and it can be used to many different kind of cancer. It can be used as its own, like the cellular therapy alone or gene correction therapy alone, or adoptive therapy with other types of traditional treatments of cancer such as chemotherapy or radiation therapy. There are different processes like blocking the process to protect the cancer cell. Some gene therapies block processes that cancer cells use to survive. For example, most cells in the body are programmed to die if their DNA is damaged beyond repair. This is called programmed cell death or apoptosis. However, cancer cells block this process so they do not die even when they are supposed to. Some gene therapy strategies aim to reverse this blockage, and scientists hope that these new types of treatment or biotherapy will make the cancer cell die specifically. It can be used by the altered viruses, like the different viruses structure you alter, you put your gene in that viruses to infect and kill the cancer cells. Researchers are working on many ways to change these viruses so they only target and kill specifically cancer cell and leaving healthy cells alone. This sort of treatment uses the virus to kill cancer cell direct rather than to deliver genes only. So it is not the cancer gene therapy in the true sense of the word. In fact, it is a viral therapy for the true sense. However, researchers refer it as gene therapy many times. One example of this type of research uses the cold sore virus like herpes simplex virus. The changed virus is called Oncovax, which is being used as biotherapy. It has been tested in early clinical trials for advanced melanoma, pancreatic cancer, head or neck cancers. There are many adoptive cell therapies which are being used for the biotherapies, like adoptive cell transfer therapy, adoptive T cell transfer therapy, adoptive dendritic cell transfer therapy, and adoptive NK cell transfer therapy. These all kind of cells being expanded in vitro by using the IL-2 and then infusing back to the patients. Many times these cells are adopted as autologous. It is better to use the autologous cells, expand and then infuse back to the same patient. And these adoptive cell therapies are being used as biotherapy of cancer many times. I will give you example of the total PBMCs or T cell isolation from the patient, expansion and then giving as the biotherapy. These biotherapy emerged method of tumor clearance aiming to afford more effective and selective targeting of tumor cells by recruiting and facilitating and existing tumor specific immune response, because the total PBMCs or the T cell expanded will circumvent many immune evasion mechanisms. T-cell immunotherapy or biotherapy for any cancer is maturing through decades, and worth of this effort it's being used now recently in many experimental and clinical trials. Various total tumor cell biotherapy techniques are associated with the T cells and have been developed during the last 2 decades and brought excellent outcomes. I will also talk about how to gene correction therapies. The genes that are damaged in the body, those genes should be corrected. Genes that encourage the cell to multiply, known as oncogenes, it should be knocked out. Genes who stop the cell multiplying, like tumor suppressor genes, genes which repair other damaged genes, it should be encouraged by the biotherapies or gene correction therapies. Gene mutations or gene at fault develop the cancer. Many gene changes that may make a cell become cancerous. These gene mutations or gene changes are caused by environmental or lifestyle factors such as smoking, alcohol, or the different lifestyle factors. Some people have inherited faulty genes which increase their risk of particular type of cancer. Some people have inherited damaged genes which is almost prevalent in 2 to 3% in every 100 persons with the cancers. So these gene defects should be corrected by gene correction therapies, and then gene correction should be allowed to do in the cancerous patients. There are, other than the gene therapies or the gene corrections, in the tumor mass there are cancer stem cells. There are now therapies which block these cancer stem cells. What is the role of these cancer stem cells? Cancer stem cells have unlimited self-renewal characterized by tumor heterogeneity. It is exclusive in vivo tumorigenicity. They are resistant to chemotherapy. So cancer stem cells have the property of self-renewal, and most of the time one cancer cell make the spheroid or cluster of cancer stem cells, and which forms the recurrence of the cancer or generate the bigger cancers. Mainly standard chemotherapy or radiation therapy or cancer therapies are being used. However, they leave the cancer stem cells, and these leftover cancer stem cells again make the tumor mass, and tumor recurrence is happening. Now we said that blocking the cancer stem cells. So these blockage of specific cancer stem cell therapies have come up. With the antibodies, these eradicate these cancer stem cells, and now the cancer stem cells will be eradicated, and there is less chances for the tumor recurrence. Biotherapies also target the angiogenesis in the cancer. What is the angiogenesis now? To grow the tumor, it depends upon new blood vessels growth, which involves three steps: angiogenesis, vasculogenesis, and intussusception. Angiogenesis is the formation of new blood vessels in the tumor area, and these new blood vessels are making from the pre-existing one vessels. Angiogenesis consists several steps like endothelial cell proliferation, because they make or they help in process of angiogenesis. Endothelial cells make migration. There is basement membrane degradation or new lumen organization. Now the growth of tumor depends on new blood vessels, and these new blood vessels are very important, and there are biotherapies that if to target these angiogenesis step, if there won't be any blood supply or the angiogenesis in the cancer, tumor mass may reduce without angiogenesis. That without angiogenesis, how the solid tumor grows? It grows less than 2 to 3 millimeter. If there are endothelial cell release angiogenesis regulators, if there is a blockage of these regulators like vascular endothelial growth factors, basic fibroblast growth factor, transforming growth factors, if there is blockage, then the tumor mass will not grow more than 2 to 3 millimeter. However, with angiogenesis and with these growth factor, tumor mass will grow more than this. In addition to angiogenesis or the presence of angiogenesis factor, there is activation of oncogene or loss of tumor suppressor gene. However, these gene correction I have already mentioned in the previous slides, so I am not going to talk in the angiogenesis subject. So the therapies are being used for anti-angiogenesis, and anti-angiogenesis has been regarded as a target for best cancer therapy. That how tumor mass generates the FGF, VEGF and TGF. These work autocrine or paracrine marrow or endothelial cells. Endothelial cells will help in migration, they will proliferation, and they make the tube formation. From in situ tumor, the cells, now the tumor become invasive tumor, and it leads to even metastasis also. How the anti-angiogenesis gene therapy help? Anti-angiogenesis gene aberration related therapy may improve overall survival in patients. In the recent study, it is mentioned that concurrent KRAS and TP53 hotspot mutant make cancers. If the therapy is given, so anti-angiogenesis therapy works. Beside modulation of angiogenesis, concomitant gene aberration related therapies may improve the treatment of KRAS positive TP53 positive mutant cancer. Therefore, anti-angiogenesis therapy is important. I will explain you what is the prodrug gene therapy. Sometimes some type of gene therapy deliver genes into the cancer cell that allow the cells to change drugs from inactive form to active form. The inactive form of the drug is called prodrug. After giving the carrier containing the gene, the researchers give the patient this prodrug. Now the prodrug may be a tablet or capsule which is swallowed, or it could be delivered into the bloodstream. This prodrug circulates in the body and does not harm normal cells. It only affects the cancer cells. But when it reaches the cancer, it is activated by the gene and the drug kills the cancer cells. So this is called as prodrug gene therapy. There are new methods which are novel and promising biotherapies like microRNAs based promising biotherapies. MicroRNAs as potential non-invasive biomarkers and tool to accurately identify, predict and treat many cancers or many diseases. What are microRNAs? MicroRNAs are short non-coding RNAs which are involved in critical biological processes. Deregulation of these microRNAs are well documented in wide range of human diseases. However, overexpression or downregulation of microRNAs affect cellular pathways that contribute to the cancer or tumor progression. Recently, LET7, MIR10B, or microRNA 21, 34, 221 are promising targets to develop microRNAs-based therapy for human malignancies. Role of microRNA 520 also developed in regulating EGFR expression in colorectal cancer. Tumor suppressor role of many of microRNAs including MIR520A in colorectal cancer regulate the EGFR expression. It is maybe the anti-MIR520A will be the novel molecular therapeutic target for colon cancer. Strategies can be used to reduce or restore the microRNA's expression in human cancer and can be used as therapeutic implications. However, little bit I will tell you about new discoveries. New discoveries of anti-LTBR signaling as biotherapies. What are the LT means? These are surface lymphotoxins which are called as LT, and surface lymphotoxins are heteromeric complex of LT alpha and LT beta chains which binds to LT beta receptor, a member of tumor necrosis factor family of receptors. LTBR signaling induce cell death via TNF or FAST receptor. LTBRs are in the soluble form also, and recombinant LTBR or the LT alpha beta expression was cytotoxic to human adenocarcinoma cell lines. These all discoveries was in the bench of the laboratories, but now that these discoveries can be used as biotherapies. There is anti-angiogenesis cancer gene therapy strategies. One of strategies RNA interference. We have to reduce the angiogenesis in the cancer and it is called anti-angiogenesis therapy. RNA interference is the sequence-specific gene silencing induced by double-stranded RNAs. Introduction of 21 to 23 small interfering RNAs of the nucleotide can knock out the expression of particular gene, and these RNA interferences therapy is being used in the cancer. A recent review written by different authors, Ezequiel et al., pointed out that siRNA against expression of vascular growth factor receptor can reduce tumor volume by blocking angiogenesis. Another known and co-workers also described a method that can suppress the expression of VEGFR-A at both transcription level and post-transcription level by combination of zinc finger protein and siRNA. Another authors, Gondi and co-workers, have demonstrated the potential application of RNA interference in gene cancer therapy by inhibiting not only angiogenesis, not only in vivo, in vitro, also in different cancer, especially in vitro human glioma cell models. Furthermore, it is possible to include more than one anti-angiogenesis siRNA into a single retroviral vector. Because small size of these siRNAs, you can fit the 2-3 siRNAs in one vector, which can inhibit the not only angiogenesis, then other pathways also, and it is used as multiple pathways blockage. Targeting angiogenesis is a promising approach in suppressing tumor growth and metastasis. Due to need of this technology, long-term administration of inhibitors, gene therapy has become an alternative, which theoretically ensures a sustained availability of the anti-angiogenesis agents. Up till now, researchers on anti-angiogenesis cancer gene therapy remain in preclinical stages. It is anticipated that when better vectors are developed for anti-angiogenesis, the molecular mechanism of angiogenesis or inhibitors against tumor growth are better understood. Clinical trials will be undertaken after the development of better vectors for anti-angiogenesis therapy in future. Target the LTBR signaling, it reduces the cancer. In the animal model of liver cancer, it is being used that hepatocyte express the LT alpha 1 beta 2 and LTBR receptors. When the two hepatocytes join together, it activates the NF-kappa B pathway and secretes many chemokines like CCL2, CCL7 and many chemokines. These chemokines induce the recruitment of many molecular NK cells, macrophages T and B, and make these liver cancer at the chronics. However, if the LTBR signaling is blocked or antibodies of LTBR is being used in the animal model, these LTBR signaling will stop and it won't recruit the pro-inflammatory molecules or the cells which can make the cancer. So liver cancer can be reduced by blocking or by targeting the LTBR signaling. However, this discovery is still in the animal models. It can be utilized further in the human studies. Side effects of biotherapies? Many. There is pain, swelling, soreness, redness, itchiness whenever you infuse or wherever the infusion or injection of cells is given. It also creates the flu-like symptoms like fever, like chills or weakness, dizziness, nausea and vomiting. There is muscle pain, fatigue and headache also. However, many biotherapies cause the fatal hypersensitivity and their allergic reactions. Urine infection or other minimal infections are very common. However, the very worst side effects are generating the secondary cancers in the body or the total organ damage. So students, let's now summarize what we have learned in this module. Biotherapy treatments can aid your immune system either directly or indirectly. Biotherapies aid to fight cancer cells. Indirect assistance of biotherapies also add into prevention of cancers. Many biotherapies, including adoptive transfer of cells, gene therapies, gene correction therapies, antiangiogenesis therapies, stem cell therapies are popular. Now, new signaling molecules like LTBR signaling molecules I have discussed are in the discovery mode and standardized in the animal models. However, in the clinical trials, they are still not have come. Thank you so much.